The manipulation of yeast cells to lower ethanol concentration in wine. Yeast cells are essential fungi used in the process of fermenting sugars from grapes to ethanol in winemaking. Saccharomyces cerevisiae is a specific strain of yeast most commonly used among winemakers as it has a higher tolerance to alcohol than other strains. Ethanol is produced from yeast during fermentation as part of the final stages of glycolysis. Pyruvate is reduced by the catalyst enzyme pyruvate decarboxylase to form acetaldehyde, and then further reduced by alcohol dehydrogenase, ADH, to form ethanol. During this breakdown of sugars, glycerol is also formed. There are many reasons to want to lower the ethanol content in a wine. High ethanol in the nose and palate can mask desirable characters in a wine and throw it out of balance. There are also issues in certain countries surrounding wine tax, as the higher the alcohol content, the more expensive the taxes are. Another reason to attempt to lower alcohol consumption in the hopes to lower alcohol abuse in society. Temperatures in many vineyard environments are on the rise, resulting in higher sugar content in grapes, affecting their fermentation properties. This impacts winery productivity, as ferments with higher sugar content have a higher chance of becoming stuck or sluggish, resulting in a lower quality wine containing off characteristics. Current research shows that most of the cost-effective and simplest ways of lowering alcohol content in a wine are to redirect the yeast glycolytic pathway away from ethanol production. This is done by changing complementary cellular processes within the ethanol fermentation that maintain a redox balance, and in particular the NAD+, NADH balance. Strategies that have been shown to work in this regard include genetic modifications targeting the gene encoding enzymes involved in maintaining redox balance during sugar fermentation, enzymes connected with the fermentation pathway, and proteins that modulate glycerol export from the cell. Through the glycolytic pathway of converting glucose fructose to ethanol and glycerol, the bulk of the NAD+ is reduced when glyceraldehyde 3-phosphate is co-oxidized to 1,3-bisphosphoglycerate. Cells must recycle the reduced form of NAD+, as it is essential for glycolysis to proceed. If the balance of NAD+, NADH is not maintained, the glycolytic flux will decrease, potentially leading to the depletion of ATP energy charge, which could be lethal for the cell. A large proportion of the NADH produced through glycolysis is subsequently oxidized during the formation of ethanol. Although there are other minor pathways displayed in Table 1 which contribute to the redox balance, in normal functioning yeast, the reduction of acetaldehyde to ethanol yields the greatest amount of NAD+ and provides the importance for the production of ethanol to maintain redox balance within the cell to continue glycolysis. Genetic modifications have been trialed to analyze the effects of the yeast metabolism through regulating genes within the glycolytic pathway. Upregulating the GPD1 and GPD2 genes responsible for reductively converting dihydroxyacetone phosphate, DHAP, to glycerol 3-phosphate, G3P, which is subsequently dephosphorylated to glycerol by glycerol 3-phosphatase. Overexpression of this gene observed a decrease in ethanol production up to 35% and an increase in glycerol up to 584%. Although a reduction in ethanol is achieved, a balance of NAD+ and NADH ratio is not maintained, resulting in a production of undesirable products produced such as acetate to resurrect the redox balance. Another manipulation of the central carbon metabolism is the reduction of the PDC gene. Within Saccharomyces cerevisiae, there are 3 PDC genes, PDC1, 5, and 6. These are upregulated by the transcription factor PDC2P. It has been established that only one PDC1 and PDC5 are known to be active through fermentation. If deletion of all 3 PDC genes occurs, then the yeast is unable to grow in a sole carbon source of glucose. with an excess of NADH produced, shifting the redox balance. Although when PDC2 gene was deleted, 19% of residual pyruvate decarboxylase maintained, which supported growth on glucose, and a reduction of ethanol, 28%, and an increase in glycerol, 366%, was observed. Alcohol dehydrogenase, ADH, is encoded by specific genes ADH1, 3, 4, and 5. They play an important role in the final steps of fermentation by catalyzing the reduction of acetaldehyde to ethanol. This reaction requires NADH, and in the process oxidizes it to NAD+, which helps maintain the cellular redox balance. If any of the ADH genes are deleted or impaired, there will be a substantial decrease in the amount of ethanol concentration, but also a decrease in the oxidation of NADH. This redox ratio can be brought back to the equilibrium by an increase in glycerol production. If a yeast strain is missing ADH1 gene, it will have impaired ethanol synthesis and a moderate increase in glycerol synthesis. However, a yeast strain possessing a mutation of ADH1 gene resulted in a third of ethanol synthesis being produced and a significant increase in glycerol synthesis. In conclusion, it has been demonstrated that it is possible to genetically modify yeast to lower alcohol concentrations. However, undesirable products are formed in the process. It is possible, with some further GMO-based research, that these unwanted products could be manipulated or eliminated, but the general public have little acceptance of GMO products. The short-term answer may lie in knowledge gained from GMO-based studies to implement the strategies for somehow generating non-GMO yeasts that are commercially viable in reducing ethanol concentrations.